Our topic today will be on the replication of DNA. And in order to understand this, we need to review the structure of DNA first. So what is DNA? Deoxyribonucleic acid is the double helix hereditary biomolecule in cells that stores genetic information needed for the different functions of the cells, such as cell growth and reproduction. Since DNA is a molecule that carries most of the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms, it must really be an amazing feat to those who discover and study this miraculous molecule. Which brings me to this point, the history of DNA. Contrary to popular belief, James Watson and Francis Crick did not discover DNA. DNA was first isolated by the Swiss physician Friedrich Miescher in 1869. He was studying the samples collected from used bandages in hospital when he discovered a microscopic substance in the pus of the bandages. He bathed the cells in warm alcohol to remove the lipids and then set enzymes loose on them to digest the proteins. What was left was some stuff that he knew might be a new kind of biological substance, and he named it nuclein, which later was called nucleic acid. Rosalind Franklin, a young biophysicist, was the one that deduced the basic dimensions of DNA strands, and that the phosphate on the outside was of a helical structure based on the photographs that were taken of DNA at that time. At the same time, James Watson and Francis Crick theorized the double helix model of the DNA structure, and it was through Franklin's work that they saw proof their theory. They got to publish their work straight away, and after that it was all sunshine and rainbows for them. James Watson and Francis Crick were awarded the Nobel Prize in 1962, while it was Franklin's work that led to her death at 37 from ovarian cancer. It was Albert Castle that isolated the DNA 5' nucleobases in 1878, and in 1919, Phoebus Levine identified the base, sugar, and phosphate nucleotide units. The structure of DNA. DNA is a double helix structure made up of two polynucleotide strands that are anti-parallel of each other. The polynucleotides are made up of nucleotides. A nucleotide is a molecule which consists of three parts: the deoxyribose sugar and the phosphate, which makes up the sugar-phosphate backbone, as well as a nitrogenous base containing either adenine, thymine, cytosine, and guanine. This brings us to our main point, the DNA replication. It is the process whereby copies of the DNA are made. DNA consists of two strands, so they will be unwound, and enzymes will read each strand and synthesize a new strand respectively. This results in two DNA molecules. So where does DNA replication occur in cells? In eukaryotes, it occurs in the nucleus, while in prokaryotes, it occurs in the cytoplasm. DNA is the ultimate director for cells, and it codes for all your traits. So when you have a really important molecule like DNA that is ultimately responsible for controlling the cell, it would make sense that when you make another cell, like in mitosis, you would also have to get more DNA into that cell. So the purpose of DNA replication is that it is needed to produce the same copies of the DNA molecule. So when the cells divide, there will be a copy of the DNA in each of the new cells formed. DNA replication is a semi-conservative replication, which means that it will produce two copies that each contain one of the original strands and one new strand. How do we know that DNA replication is semi-conservative? An experiment by Matthew Meselson and Franklin Starr, called the Meselson-Starr experiment, along with help from Mason McDonald in 1958, supported the fact that DNA replication is semi-conservative. They used an isotope of nitrogen of which had an extra neutron, making the DNA heavier to distinguish between the parent and the newly copied DNA. DNA replication involves three main things. First, the unwinding of the DNA. Secondly, pairing the bases in each strand with new bases to form new complementary strands. And finally, producing two new DNA strands that exactly duplicate the original DNA. So, what are the enzymes involving in DNA replication? Firstly, there is the DNA helicase, DNA polymerase 1, DNA polymerase 3, DNA ligase, primase, and lastly, RNase H. How does the DNA replicate? Firstly, the DNA helicase will unwind the double helix at breakneck speeds, slicing open those loose hydrogen bonds between the base pairs and form the replication fork. Next, a primase will form a RNA primer with the free 3' OHN each for both synthesis of the leading and lagging strands of the DNA. This is important because no DNA polymerase can synthesize the DNA de novo, so it needs a primer for it to hook onto so it can start building the new DNA chain. DNA polymerase 3 will then start the polymerization of nucleotides in the continuous 5' to 3' direction for both leading and lagging strands. The leading strand is made continuously in a 5' to 3' direction, but the lagging strand is made discontinuously in many short Okazaki fragments. Each of the new Okazaki fragments are separately primed, polymerized, and ligated in the 5' to 3' direction. At intervals, the template for synthesizing the lagging strand will signal the primase to make RNA primers. DNA polymerase 3 will synthesize DNA from the last primer towards the previous primer to generate those Okazaki fragments. RNase H will remove majority of the RNA primer except the terminal ribonucleotide. Then DNA polymerase 1 will perform nick translation and remove the terminal ribonucleotide and fill in the gaps left by the primers with DNA nucleotides. Finally, DNA ligase will seal off the remaining nick between the 5' monophosphate and 3' OHNs of the newly synthesized precursor fragments to form a continuous lagging strand. Now, the fidelity of DNA replication. The survival of an organism depends on the accurate duplication of the genome, and thus, fidelity is important. Any mistake during replication will result in a permanent mutation. The fidelity of DNA replication can be traced to 3 distinct activities. 1. The accurate selection of nucleotides. 2. Immediate proofreading. And 3. Post-replication mismatch repair. Although DNA replication is a truly amazing biological phenomenon, it is not always perfect. While most DNA replicates with fairly high fidelity, mistakes do happen, with polymerase enzymes sometimes inserting the wrong nucleotide or too many or too few nucleotides into a sequence. When this happens and were not corrected by the repair enzyme's mechanism, they become permanent mutations. Now, let us look at some of the different types of DNA mutation. Firstly, there is a substitution. A base is exchanged for into another base. Second, the insertion. Extra base pairs are inserted into the DNA. Third, deletion. A section of DNA is lost. And lastly, frame shift. Insertions or deletions that cause the gene to no longer be read as the nucleotides are arranged in threes as codons. And thus, this concludes our presentation on the topic of DNA replication. Thank you.